So far in looking at genetics, we've looked at inheritance of a single trait and with the simple dominance or recessive trait. Now we're going to start looking at the inheritance of two traits. So a little bit of review of chromosomes, and particularly homologous chromosomes. They're inherited as a pair, and again, this is for a diploid organism such as ourselves. We have one member from each parent, one from the mother, one from the father. And because they're homologous, they have the same length and similar genes, and they're going to be in the same order, same centromere location. That's where we get homologous from. During meiosis, each gamete receives one member of each pair of homologs. This is why during meiosis we get haploid cells from diploid cells. Now the homologs separate independently. When we're looking at that concept of independent assortment, it doesn't matter which member of a pair goes into which gamete. And so that means, and we'll see this momentarily, it's not as though all of the chromosomes from mother go to one cell, all from the father go to another, or that they make sure that there's an equal balance. It's a little bit like a flip of a coin. So we can assume that the alleles for two genes on these homologs in the following examples. So a little review of meiosis, and when we're talking about independent assortment and all those concepts we were just covering, if we're looking at a mother cell, and this is a diploid cell in which 2n equals 4, we have these pairs of homologous chromosomes, so represented by S's and W's. So we're looking at the concept of widow's peak or straight hairline, so hairline there with these W's, or short fingers versus long fingers with the uppercase S and lowercase s. So you can see that there's the same kind of information, whether it's finger length or hairline, but color-coded, looking at which is inherited from a separate parent. So during meiosis 1, after we have duplicated chromosomes with sister chromatids, that's represented here. These two are sister chromatids, this is a pair of sister chromatids, etc. So whenever they line up during metaphase 1 of meiosis 1, they could line up either this way or that way. And keep in mind here, we're just looking at two different chromosomes here. So with humans, we have 23 pairs, so this would be a lot more complicated. During meiosis 2, after these homologs have gone to separate sides of the cell during anaphase and telophase, then we have either this result or this result. At the end of meiosis 2, when we have daughter cells that are formed, we could either have these 4 haploid cells or these 4. So, since we don't know how these homologs arrange themselves independently during meiosis 1, we're not sure for the resulting gametes which alleles they will carry. Looking at this figure is the long explanation of how we're going to get these combinations of alleles in the following problems. So let's look at a two-trait cross. If we have a dihybrid cross that's going to be a hybrid for two traits, we're going to use this to test the law of independent assortment. So for the example of how we get a dihybrid in the first place, if we have a person who is homozygous dominant for widow's peak and short fingers, that's represented as big W, big S, big S. Notice here too that for two traits we have 4 alleles, because we have 2 for one trait, 2 for the other trait. Since you have homologous pairs, you have information from the mother and the father. Okay, so we have homozygous dominant is going to be crossed with a person who has straight hairline and long fingers. That's homozygous recessive. So here's our cross. Homozygous dominant for 2 traits, homozygous recessive for 2 traits. So law of segregation, no matter how we combined these 2 traits, we're only going to get dominant alleles, so big W, big S. Likewise, the gametes for the homozygous recessive parent is going to be little w, little s. whenever we combine that for potential of the offspring, it's all going to be this dihybrid of the big W, big S, little s. All offspring have the same phenotype. So if we're looking here, this is the genotype. What's the phenotype for that? This is how they physically look. That's going to be widow's peak, represented here since we have a dominant allele present, and short fingers represented here since we have a dominant allele present for short fingers. This phenotype is called a dihybrid because the individual is heterozygous for both hairline and fingers. So this is representing the parental generation, homozygous dominant for widow's peak here, and short fingers crossed with the homozygous recessive of straight hairline and long fingers. So showing the parental gametes here, and how all offspring for the first filial generation would be dihybrids. And so this is the genotype here, phenotype being widow's peak and short fingers. So if we had a two-trait cross crossing two dihybrids, what gametes are possible? This is where we look back at the law of segregation and law of independent assortment. Law of segregation states that each gamete gets only one letter of each kind. So that's looking at a single allele for each trait. Law of independent assortment states that all combinations are possible. In order to get that, we're looking at every combination of W's and S's here. If we work this outer to inner, so if we look, we can have a big W with a big S. We could have big W, little s. Okay, so we've got all of the big W's taken care of, right? Now if we work inner, we could have little w, big S, or little w, little s. That's what's represented here. These are the gametes that are possible from a dihybrid. So far, we've only worked Punnett squares that were 2 by 2 with a single trait. Now that we have 4 possible gametes with 2 different traits, we need a 4 by 4 square. So this one's already worked out for you. So the gametes that we just figured out, shown here on the top and along the sides, since we have parents that have the same genotype. Working out a Punnett square like this is a little tedious, but you're going to get a predictable pattern every time you have a dihybrid cross. So this is color-coded for us. So we're looking at genotypes represented by these alleles, and we have the phenotypic ratio given here. And this is also color-coded, so we're saying that 9 of these 16 have a widow's peak and short fingers. How they're figuring that out is in order to have a widow's peak and short fingers, so this would be dominant characteristics, we could have the genotypes with all capital letters, right? We could also have a combination where we have one big W, one little w, big S, right? That would also give us widow's peak and short fingers. We could have another dihybrid, right? Or we could have big W, big S, little s. All of those genotypes have the same phenotype. And so if we look here, that's represented here. Okay, so got that one taken care of. And so as you're going through to figure out phenotypes, you'd go square by square to figure out who has a widow's peak and short fingers, who has a widow's peak and long fingers, straight hairline short fingers, and straight hairline long fingers. Our next phenotype is having widow's peak and long fingers. So that's represented by these gray squares here. So in order to have widow's peak and long fingers, we could have the genotypes, big W, but the only way to have long fingers is to have those two recessive alleles. So we could have that combination. They could also be heterozygous for one trait and homozygous for the other. So that's what's represented by these three squares. Looking at the third phenotype that's possible, straight hairline and short fingers, the only way to have straight hairline is to have two recessive alleles, but to have short fingers, we could either have two dominant alleles or one of each. That's what's represented by these three here. The last phenotype we have is straight hairline and long fingers. Those are two recessive traits. There's only one genotype that's possible for that. That is represented by this last square. Every time we have a two-trait dihybrid cross, there's an expected phenotypic ratio. So here we have 9 to 3 to 3 to 1, where the 9 are the two dominant traits, in this case widow's peak and short fingers. 3 is one dominant trait, one recessive trait, so widow's peak and long fingers. The other 3 is just the opposite of that, so we have the other recessive trait and the dominant trait, straight hairline, short fingers, and the 1 is two recessive traits, straight hairline, long fingers in this case. You can also use probability laws and the product rule to get these same results as we worked with the Punnett square. To calculate the probability of having a widow's peak is 3 out of 4. The probability of short fingers is 3 out of 4. Probability of straight hairline, 1 out of 4. Probability of long fingers, 1 out of 4. So in order to get those 4 different phenotypes that we had, widow's peak, short fingers, we multiply those probabilities. And so again, 3 out of 4 times 3 out of 4, we get 9/16. Widow's peak and long finger, we get 3/16. Straight hairline, short fingers, 3/16. Straight hairline, long fingers, 1/16. So you can see we still have this 9 to 3 to 3 to 1 ratio. It's just worked out with probability laws instead of a Punnett square. We could also work this out with freckles and finger length. So this shows that same dihybrid cross, only this time instead of hairline, we're looking at the presence of freckles or no freckles. But notice how we get that same pattern and same ratio. So whenever you work this out, you get 1, 2, 3, 4, 5, 6, 7, 8, 9 with both dominant traits. 1, 2, 3 with one dominant trait, the other recessive, and we get 1, 2, 3 with recessive trait, dominant trait, and this final 1 out of 16 here with both recessive traits.